Good evening, ma'am.
Good evening, sir.
Good evening, ma'am.
Sir, you can start now.
Yes, ma'am. Thank you. Thank you so much, ma'am. Good evening, my dear students. Good evening, Ajay.
Good evening, sir.
Good evening, Kana. Okay. So, once again, you are all are warm welcome to the wonderful session of the biological class. Okay. So, last class, what we have discussed, you know a little about what is that? Lac-Operon concept. Okay. The Lac-Operon concept, what are the content present in it? It consists of 4 genes, 4 to 5 genes there. So the one gene is called as, what is that, I gene. It is also called as regulator gene. 3 structural genes are there, Z, Y, A, and 1 operator gene, 1 promoter gene. So total, how many genes, total 6 genes also present in it. Each one, it has a specific functions. Okay. Particularly, the inducer, which one is act as a inducer? Just note it down. This is very, very important. Lactose is a inducer. Don't forget, my dear. Okay. Suppose if it is absence of lactose, absence of inducer, what happen, the mRNA, repressor mRNA produced from the DNA molecule, and it easily bind with the operator of the DNA molecule. As a result, what happen, it will not produce the, what is that? Lactoperon genes are off, completely off in nature. It will not proceed any other, what is that? Production of amino acids or enzymes or whatever it may be. Next one is, suppose if it is presence of inducer, what happen, the mRNA production takes place. The mRNA bind with the inducer. So it is not bind with the operator gene. It lose their ability. So the process, it will be possible. So as a result, what happen, the structural genes produce mRNA due to the process called as transcription. Finally, beta galactosidase, permease, and trans-acetylases formation takes place. So as a result, what happened? The lactose molecule split into glucose and galactose by the enzyme galactosidase. After that splitting of molecules, the permeases allow the molecule from the outside to inside, and also some translation process occurs within the bacterial cells. These are all are called as, what is that? Lactoperon concept. Already we discussed, isn't it? Okay. Today, we are going to discuss about the genetic code. Already we saw it, introduction only. Today, we are going to discuss about how genetics are forms, who discovered. It is also denoted as amino acid, how many amino acids present in it, how they are classified, what is the structure of the amino acids. The things we are going to discuss now. The first, genetic code. The term genetic code was given by the name of the scientist called George Camo. Don't forget, my dear. George Camo. The relations between the sequence of amino acid, the genetic code, each and every genetic codes represented 1 or 2 or 3 monolet, bilet, and triplet codons. But monolet, it does not indicate any amino acid. Triplet, it is not indicate any amino acid, and triplet codon only indicate the amino acid. Each one, it has a 3 nitrogen bases. Each 3 nitrogen bases indicate the 1 amino acids. Okay, see that the relationship between the sequence of amino acids in a polypeptide chain and nucleotide sequences of the DNA or mRNA. So DNA or mRNA is called a genetic code. Already we saw it, these things, isn't it? Okay. There are 20 types of amino acids which participate protein biosynthesis. So 20 amino acids is the 10 is essential, 10 is non-essential. Already we discussed. We'll see one by which one is essential, which one is non-essential, and we will see the next slide. DNA containing information for the synthesis of any types of polypeptides, because the DNA, it has a control, the master of cells. It controls all the activities within the cells. So the protein biosynthesis, enzyme biosynthesis, vitamins, mineral, carbohydrate, protein, lipids, whatever it may be, all the activities was controlled by the, which one, master component, master cell, which one, the nucleus. So nucleus, which inside containing what is the DNA molecules. So the DNA containing information for the synthesis of any type of polypeptide chain. The polypeptide chain, it may be a protein molecule or lipid molecule or enzymes or hormones or whatever it may be. It possesses, it has the ability to produce. Okay. So in the process of transcription, information transfer from DNA to mRNA. That means, already we studied that message, the particular polypeptide chain production, that message is transferred from DNA to RNA through the process called as transcription. In the formation of complementary N2 base sequences, then only what happened, the mRNA production takes place. Already we discussed, isn't it? Only one strand only produces, isn't it? For example, take an examples. So we will see next slide. Okay, see that one. And then generally, what are the properties that the genetic code it has? See here, the genetic code is composed of a nucleotide is triplets. What is the meaning of triplets in this part? Taken examples, A. Okay, otherwise called as G, and U. Okay, and also C. So not only this, and also AUG. So combination of others also, adenine, uracil, guanine. These all are called as what is that? Triplets, because it's contain 3 nitrogen bases. Each one it is a AUG, it is denoted as a methionine, isn't it? Methionine. Okay. So met, simply we may call as methionine amino acid. So the AUG is denoted as which one? So methionine. Don't forget, my dear. This is very, very important. See. So AUG, methionine. Likewise, each one it has a specific amino acid. We will see. The code is non-overlapping. Already we discussed. This means that successive triplets are read on order. Each nucleotide is part of only one triplet codon. So each one it has only one triplet codon is there. The genetic code is unambiguous. Why it is called as unambiguous? Because one side acid is there, another side is the base is also present. It is. So both. Each codon specify a particular amino acid and only one amino acids only. So this is the major important ones. So particularly each one it has a mention as a. So for example, AUG, only methionine. Is it? It indicates the methionine amino acids encoded by a triplet code. And some cases in genetic code is degenerated. It contrasts some amino acids can be specified by more one codons. This is also a exceptional. Okay. See the next one. The code is nearly universal. Why it is called as universal? Worldwide, they are following this type of what is that? Codon. Okay, that's why the code is nearly universal. Almost all organisms in nature, from the bacteria to human beings, so the lower to higher organisms, you see exactly the same genetic code. So we will not change it. So for example, lower animals, bacteria, the DNA contain, the genetic material contain genetic code. It is not different compared to the human being. Both are whether lower or higher, whether plant or animals, whatever it may be, they are all what is that? Exactly the same genetic code. AUG is mentioned as a methionine for all the categories. Okay. The rare exception includes some changes in the code in mitochondria and in a few protozoans. This is also very rare. Okay. The genetic code specify 20L. So what is the meaning of L? Already told you. This is denoted as a levorotatory of alpha amino acid. You know about the protein molecules are made up of more than one amino acids. But how many amino acids present in it, my dear? Ajay, how many amino acids there?
Sir, 20.
20 amino acids, sir. Very good, sir. So all are which type? Which type of amino acids?
Sir, 20L.
L. And alpha amino acid. And also beta amino acids also there. That's why. See the structure, you can easily identify. See that structure, you can identify the things. This is called as L-alanine. They have given as a alanine is the basic amino acid. D-alanines. The L denotes levorotatory. See the structure. The center, there is a carbon atom is there. So here hydrogen is also connected. One side there is an acid group and opposite side amino group. Okay. So CH3, this is the methyl group, isn't it? That's why it is a simple amino acids. See dextrorotatory. This is called as dextrorotatory. See this one. Okay. See this one. See the hydrogen where it is in the left side. Okay. So this one is right side. This one is left side. And also see that the CH3 is present here. H is present here. Same as it is. So this condition is D-alanine. This is called as levorotatory. D-alanine is called as dextrorotatory and also levorotatory. So this is a negative charge. This one is a positive charge. That is a X. Okay, see the next slide, you can easily identify. This L-alanine. See that NH3, H. But here what happened, it's also opposite manner. See that H is present near first. In this portion, H is here. So in this portion, what happened, the NH3 is present in this side. So others are same. This one is called L-alanine. This one is called D-alanine. See that L-alanine, D-alanine. The structure is also similar. Okay, these are the things. Next, we will pass on to the next one is the essential and non-essential amino acid, my dear students. Very, very important. What is by essential? What is by non-essential? The required for the body process. Take an example. Any one of the protein molecule, other than the protein molecules, they need essential amino acids. But the essential amino acids not synthesized in our body. It is supplemented by where? In the outer sources. Don't forget. So what are the essential amino acids given here? See that these are the essential amino acids. Histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. See, they got tryptophan, phenylalanine. So these are all what is it? Aromatic amino acids. Okay. So these are all what is that? Aromatic amino acids. So these are all considered the getting known as essential, but did not synthesize in our body. Supplementation from the outer sources by food only, whether it may be non-veg or veg. So this is the non-essential. Non-essential. It is synthesized in our body. That is very, very important. Non-essential, synthesized in our body. But these are the non-essential amino acids. See that alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, and proline. These are the amino acids or non-essential. So total 20 amino acids. 10 is essential, 10 is non-essential. Okay. Some adult may be able to synthesize histidine on their own. Some of the category, histidine. So not histamine. Don't forget, my dear. Okay. See that one. Essential amino acid, non-essential amino acid, conditionally essential amino acid. Okay. So conditionally. Sometimes. Okay. Arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So non-essential alanine, asparagine, aspartate, glutamate, glycine, serine, and tyrosine. These are the non-essential amino acids. Conditional essential amino acid. What is the meaning of conditionals? Compulsory. Okay. There is a if like that. Yeah. Question like that. Cysteine, glutamine, hydroxyproline, proline, taurine. So this is the another one. This is also a another type of amino acids. Okay. So these are the important. Here what they are asking, the questions is, which one is the basic amino acid? They ask the questions. Okay. Which one is the basic amino acid? Will you please, anybody else? Which one is the basic amino acids? Basic amino acid. You just note it on, my dear student. Aromatic amino acid. Tricarboxylic. Okay. And sulfur containing amino acids. So these all are very, very important. You have to split the amino acid structures according to their nature. They may be divided into so type: monocarboxylic, bicarboxylic, tricarboxylic, and also there is an aromatic ring structure containing a sulfur containing amino acid. Like that, you have to differentiate. There are, I think, 5 to 7. It may be classified according to their nature and the structure. Okay. So next. So we'll pass on to the next one is how the genetic codes of formation takes place. How it will be possible. If a genetic code is a singlet, that is codon is the combination of one nitrogen bases, then only 4 codons are possible. So only one codon means A, adenine, C, cytosine, G, guanine, and U only. These are insufficient to code for 20 type of amino acids. So these are not sufficient because the single code, so 4 power 1, that is equal to 4 codon only. So A, C, G, U. I have given you only one 4 power 1. 4 codon only is the single number of n. 4 into n. So the number is 1. So you will get 4 codons only. So it is not sufficient for the formation of 20 types of amino acids. So it is not possible. Okay. So what about the next one? See that if the genetic code is 2, what happened? 4 power 2, you will get 4 into 4, you will get 16. Suppose you add like this, AA, okay, CG, GG, UU. So it is also comes from, 16 only it comes. Okay. So it is not sufficient for the 20 amino acids. Once again, repeat for you. Suppose if it is 1, only 4 codons are possible. Suppose if it is 2, so for example, addition of one more, adenine, like that. Okay. The 20 amino acids, it code for only 16 only. See that? 4 amino acids that you can change it, whatever it may be, AA, AC, AG, AU. Okay. So you will get only the 16 molecules only. So it is not sufficient for the 20 amino acids. Here the amino acids will be high compared to the codon. So next, so we come to the conclusion. What is the triplet codon? It's a very, very comfortable and most reliable method. Okay. So the main problem of genetic code was to determine the exact number of nucleotide in a codon which code for 1 amino acid. It's code for 1 amino acids. So there are 4 types of nitrogen bases in mRNA. Already you know about adenine, uracil, guanine, and cytosine for 20 type of amino acids. It is sufficient for protein biosynthesis or translation process. Okay. See the triplet codon. 4 power 3, that is equal to 4 into 4 into 4. You will get 64. 4 into 4, 16. 16 into 4, 64. So this is sufficient for 20 amino acids. Okay. See that one. Suppose if you want to more information, just to go to the link I have given here in YouTube. Okay. How the genetic information, so genetic code formation takes place. Okay, my dear students. Okay, for the next one. So these are the, what is that? Genetic code formations. How many genetic code is also present in here? I will show you how it will be. Okay, so you just listen here carefully. See the UCAG, that is a formula. UCAG. Okay, see here how it will be. I will show you in different manners. Already you know about the whiteboard. I will select. So what is the codon name? Is the UC and AG, isn't it? This is the top first codon. So this is the first one. So second one and third one, because 3 codon is very, very essential for the 20 amino acids. So the same things is also UCAG, isn't it? The same things also repeated again. UCAG. So this is the triplet codon. You will get it. How much is the 4 into 4 into 4? You will get 64. So it indicates how many amino acids are now. Just imagine 20 amino acids. Simply AA, it's denoted as 20 amino acids. How it will be formed already, you know, but it is simple max calculation only. Just you draw a small, what is that? Part or say this one, just draw a small square only. Okay. How many codon is that? This is the first codon in this side. So you have to write the second one in this part. So third one in this part. Okay. So you have to mark it. You have to divide into 4. See here. So 1 and 2, 3 and 4. So here also you have to divide into 4. 1, 2, 3 and 4. Okay. So here you take a first one in this part. Okay. It have to take put the diagram in this part. So UC, see here, you have to change the colors, then only you can easily understand. So this one, okay, see that U C A G, isn't it? This is the first part. Second part, you have to place it here. Okay, place it in this part. Okay, change the color. So then only it will be better for your understanding. See here, U C A and G. In this box, we selected only one, okay, only one codon only. But in this last one, you have to place the 4 codon in a single box. See here in this part, you have to place a 3. See that? Here you have to place it, which one, U C A G. Okay. So this is a first one. See that one. So then only it will be better. See here. Same idea. So in this part, how will you select, first one? So the first you have to take this one. Okay. First you have to take this one, U. Okay. The first one is U. Second, this one, you have to place it here. Okay. U. And third one in this part, you have to take U. And second, again, you have to take this one, U. Again, U. You take the second one, C. Okay. Likewise, you have to frame. So how many amino acids, how many triplet codon possibilities there, you will get 64. Okay. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. 16 is, you will get it. So 4 into 4 into 4. 16 square is the, okay. How many codon is there? Already know 4 codon is there. So you'll get, how much is there? 64. Okay. From this part, in this area, just note it down. In this part, mostly what happened, so this the in this part, the second and third, and also another one. These 3 combination, what is that? Stop codon, it will come. Stop codon. In this portion, I will show you where it, and the slide, when you are seeing the slides, you can easily identify it. Stop codon is there. So already you know about the AUG is the, which one AUG is the, which one starting codon, isn't it? Starting codon. Starting codon. Okay. So AUG is a starting codon. Stop codon, how many codons comes, 3 stop codon comes. When during the translation process, 3 stop codons comes, any one, it comes, automatically what happened? Continuously stop codon, again stop codon, again stop codon means that process completely finished. So that means protein biosynthesis or enzyme biosynthesis, whatever it may be, albumin, globulin, or heparin, or whatever, in which protein synthesis the message comes from the nucleic acids completely finished. Okay. Again, it entered the name one. Are you clear in this area, my dear? Ajay?
Yes, sir.
Okay. Okay. So, okay. See here. So, see that structures, then only you can easily identify. I will show you. See here. So, this is the structures. See here, the given here, the first base is here, second base is here, third base is here. The first base, see here, UCAG. Okay. Here, UCAG. See that each one it has a complete 4 codon. UCAG, 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 like that. So when we are selecting these two, 1, 1, and also this one. Okay, we get first one, UUU. That is denoted as the phenylalanine amino acids. And also another one is a UU. Second one is called as C. So that is called as phenylalanines. Okay. And UUA, leucine. So UUG, which one is called as, what is a G is called as leucine. So how many leucines are there? See that 2, 4, 6. Already told you. Some of the nucleotides it also mention more than one amino acid. This is called as leucine. This isoleucine is also 3. Methionine is a starting codon. Just already told you. AUG is just starting codon. I told in this part. So in this part there is a stop codon. Okay. So this is the UAA, UAG, UGA is a stop codon. These three only the stop codon. Any part, you can see it. There is no any other stop codon is here. In this part only 3 stop codons comes during the formation of tabulation. And AUG is a starting codon. So only one is the methionine present and only one methionine present in it. It indicates the AUG only. Others it is not indicate the methionines. So that's why it is a starting codon. So when it the stop codon UAA, UAG, UGA comes, you know what happened? It is the process completely stopped. Okay. See, they have given us a different colors. Suppose if it is pink colors, leucine, isoleucine, valine, alanine, proline, and glycine. These are all what is that, you know, non-polar aliphatic amino acids. Okay. And the next one is what is that, you know, uncharged, polar uncharged. Which one is there? In the light blue color. See that? Serine, threonine, glutamine, and also asparagine, cysteine, and serine. These are all called as unchanged, polar and unchanged. Already told you the aromatic amino acids. What are the aromatic? It has given is a positively charged. Which one is the aromatic? The phenylalanine is the aromatic. Tyrosine is also aromatic. Tryptophan. This is also a very, very important question, my dear students. Which one is that? Which one of the following is there? Which one of the following amino acids? Aromatic amino acids. Sometimes they may ask the question. Phenylalanine, tyrosine, and tryptophan. PTT. Okay. Phenylalanine, tyrosine, tryptophan. These are all, it has a aromatic ring structure, just like a benzene. Okay. So that one. And positively charged. Which one is positively charged here? You know, there are 3 enzymes given here. One is called as histidine, lysine, arginine. So these are all what is a positively charged amino acids. And the green colors, this one is called as green color, aspartic acid. See that only one negative charged amino acid present in the total 20 amino acids. Only one is there. Okay. So these are all according to their nature and also structure aspect. They have maybe classified. Okay. Basic, what is that, you know, how it will be framed. Okay. The triplet codon, how it will be framed as a 64. And how many amino acids there? 20 amino acids, how it will be encoded. So that is very, very important, my dear. So don't forget, this is the important points. Here you must note it down. Which one is starting codon? Okay, AUG. Sometimes they may ask the question starting codon directly. Sometimes what they ask, no, AUG is denoted which type of amino acids, like that they may ask the question. Another one is which one is the negatively charged amino acids? Aspartic acids only, and then aromatic. Okay, aromatic. See that one? The phenylalanine, tyrosine, tryptophan. The negative means positive also sometimes. You note it down. LAC, LAH, HLS or LHS. What it may be? Has, HAL, HAL, HAL. Histidine, arginine, and lysine. So these are the, what is that? Amino acids. Okay, these are the amino acids are positively charged. Aspartic acid, negatively charged, as it is. So negatively charged. Okay, so this is very, very important, my dear students. So how it will be? Okay, see that one. This is also another one. The first letter here, second letter here. And this category, first letter here, second letter. Whatever it may be, you can put anywhere. It is not any rules and regulations. Okay, so this is the correct. So the first letter and second letter and third letter. The first letter U, C, A, G. All are comes from U, C, A, G. Okay, U, C, A, G. So here I told you, know, this part only. A, U, so it's the stop codon, it comes in this part only. So the third one, there is a methionine. So from this one, 1, 2, 3, there is a stop codon in the third one and fourth one. Here the third one is. In this part, the third column, the last one is the AUG, methionine. When you are putting the board, you know, checkerboard, look like a checkerboard. This is also one of the checkerboard. So you will get it that the AUG is where it comes, you know, this is the place. Okay. So this is the place, and also starting codon is also there. Okay, see the next one. Starting codon AUG, so methionine. Some of them are valine. Don't forget, this is AUG is the methionine, already told you. And GUG is also valine amino acids. Both are starting codon, depends upon their organisms. Okay, most of the cases AUG as a starting codon. Suppose instead of AUG, sometimes they were used GUG. Where is the GUG comes here? See here. G, where is it? Last one. U, where is it? First one. G, where is the last one? See that this is the part. Okay. So GUG, where is it? So G. Okay. U is where is it? The first one. G is where is it? Here. GUG. Okay. See that one? GUG valine. So GUG. So first one G, second one U, third one G. So the GG is the valine. So that was mentioned here. That is. Okay. Ambiguous codons. So AUG is the ambiguous codons. Okay. AUG is also one of the ambiguous codons. Why it is also ambiguous? Okay. See that. So the AUG code for both methionine and formyl methionines. Similarly, GG code for both methionine and valine also. So that's why it is called as what is an ambiguous, both the character. So sometimes it acts as a valine, sometimes it may be act as a methionine, interconvertible. That is the things. Punctuation codon. These are all are called punctuation codons. So what is the meaning of punctuations? AUG is the starting codon. UAA, UAG, UGA, these are all stop codon. These two are very, very important because punctuation started from the one word of the first one and end with the last one. Just that's why it is also punctuation codon. Starting is AUG, finishing with UAA, UAG, UGA. So similar to the punctuation mark. In between the word, there is a punctuation mark is there in the one side starting place as well as whether it may be a one, the single or double. Okay, punctuation mark is also there. So that is very, very important. The last one is also named, you know, stop codon. There are three. Okay, codon is it has a own specific. What is that? Bad. You know very well what that things also already we discussed. UAE, UAA, UAG, and UGA. So see here, Acre, Amper, and Opal. So these are the different names also given. So these are the name because here just see it. Here there is no any enzyme mentioned here, isn't it? See, stop only mentioned here. Next one is also same. See that stop codon, stop codon, stop codon. So when you are mentioning UAA, so it has a specific name is called as Acre. UAG, Amper. UGA, Opal. So different name has given for the stop codon. So these are the important things is there in this part. And degeneration of code or Uppel hypothesis. This is also a very, very important one, my dear. So this is a very, very, what is the Uppel hypothesis? Who proposed this theory? This was first proposed by the name of the scientist called as Crick in 1966. Okay, so Uppel hypothesis. What is that? See here. There are 64 different triplet codons and only 20 amino acids. Yes, unless some amino acids are specified by more than one codon. This is very, very important. Okay, some amino acids are specified by more than one codon. Already we saw it, isn't it? See here. Leucine 1, 2, 3, 4, 5, 6 is there, isn't it? See here, UUA, UUG, CUU, CUC, C like that. Okay. So here also the proline is also, serine also. So that has given here. Okay. Unless some amino acids are specified by more than one codon, Some codons would be completely meaningless. What is that? Stop codon here they are mentioning. Therefore, some rudimency is built into the system. Some amino acids are coded for by multiple codons. This is called as degeneracy. Okay, more than one amino acid is encoded by a different codon is called as what is a degeneracy. Don't forget, my dear students, what is by degeneracy? Sometimes they may ask the questions. Okay, that is denoted as what is that? Some amino acids are coded for by multiple codes. That is very, very important. There are 18 amino acids that are coded by more than one codons. How many amino acids are encoded more than one codon is, they may ask, means you have to answer how many questions, how many answer is there? 18 codons. Okay, 18 amino acids is essential. So this is also important. Okay, so this is the next one. See that amino acids, number of codons. Don't forget, my dear. See, yes, ma'am. Okay, ma'am. Sorry for the inconvenience, my dear students. Small intro changes here. The Whipple hypothesis is a very, very important thing. Sometimes they may ask the questions. Already know about each and every amino acid, they are having triplet code, isn't it? From this triplet code, first two letters, taken examples. First two position of the triplet codon on mRNA have a precise pairing with the base of the tRNA, anticodon T. Okay. Another one is the third position base of the codon may be ambiguous and varies according to the nucleotide present in the position. Okay, this is the very, very important. That is the, what is the meaning of this one? You know, the third one's where it is present, you know, the DNA molecule, there is a nucleotide, isn't it? DNA molecule, there is a nucleotide is there. The third one is deciding the anticodon of the, which one, you know, mRNA. So that is very, very important. This process is called as Whipple hypothesis. Okay, see here. Thus, a single tRNA type is able to recognize two or more codons differing only in the third base only, Such interaction between the third base is referred to as the Whipple pairing. So, imagine, suppose A, take an examples of A, U, G. Okay, take an examples of A, U, G is there. What is the combination of this one, you know? U, A, C. Okay, this is the third one. So, third one, you know. So, which one is the third one? So, this is the third one, isn't it? The third one is the designing of the exact pairing of the codon. So, this one is only, these two only the mRNA, isn't it? It's already in our, but the mRNA. This is the third one. What is the third base is referred to as the Whipple pairing. This pair is called as with one, you know, Whipple pairing. It's exactly match each other. So the first two is there, which one, you know, it's considered as a mRNA having precisely pair with the base of the tRNA anticodon T. The pairing of the third position base of the codon may be ambiguous and varies according to the nucleotide present in the position. So this is the base. So this is the first two is a mRNA. The last one is the Whipple pair. Okay, don't forget, my dear students. This is the deciding authority of the amino acids. Okay, in both the cases. So this one, Whipple base pairs. The first anticodon C. Okay, the third codon always will be G only. Okay, the first one is A. The last one is always will be U only. Suppose if it is U, the third codon will be, how it will be, A or G. Okay. Suppose if it is G, the first codon, so the U or C is the third one. And another one is called as I. It is also external one. UCG. 3, it will be possible. Okay. These are all are, what is that? Whipple base pairing concept. First letter should be C, means the third letter compulsory, which one, you know, C, G only present in it. Okay, see, we can see it the first letter. Suppose first letter C, the last letter is U. Okay, see here. This was given by CG, third codon base. Okay, so this is a combination of Whipple base pairs. This is also given by the name of the scientist. Already know what. A different code of archetypal code. It is found in human mitochondria, yeast mitochondria, and paramecium, PPLO, and ciliates, etc. Different type of codon is also present in it. The different type of codon is called as archetypal code. Okay, archetypal. So this is the archetypal code. So this part. Okay, so this part is a different types. It's mostly found in mitochondria, yeast mitochondria, and paramecium and PPLO and ciliates, etc. as given as an examples. Okay, other than the amino acids. Different means universal and the archetypal codes. See that the codon UAA, UAG, UGA. So these are all what is a stop codon, otherwise called as termination codon. Okay. Suppose if it is an archetypal code means what is it? Glutamine. Okay, mentioned as a glutamine. This is also glutamine. So another one is called as tryptophan. Suppose AGG is arginine is normal codon. So AGA is also arginine in the normal codon. The same codon is also indicated in where the archetypal code is a termination codon. Okay, here the termination, it is mentioned here as some specific amino acids encoded by the specific amino acids. Same process, here the amino acids mentioned in normal means here what happened, it may mention as a termination codons. Okay, so these are the important, my dear students. I think this part is completely finished. Okay, so we will pass on to the question pattern. Okay, here. The question 1, the Whipple hypothesis establishes. Okay, establishes. Which one, my dear? Economic in tRNA molecules. Economy in tRNA molecules. Peptide chain formations. Termination of peptide chains. Initiation of the peptide chains. It is not related to the peptide chains. Only it depends upon the economy of the tRNA molecules that has given here. Okay, the number is option is 1 only. Whipple hypothesis establishes economic in tRNA molecules only. It is not depends upon the peptide chain formation and termination of peptide chain, initiation of the peptide chains, because it is related to the tRNA. Okay, so this is the first one. Go to the next one. The enzyme amino acid tRNA synthetase facilitates. What is that? Facilitates. Okay, TWO. Okay, so this is the important one. Joining two neighboring amino acids on ribosomes. This is very, very important. Joining of the two ribosome in amino acids on the ribosomes. That is the point. Very, very important. Amino acyl tRNA synthetase facilitate the joining of two neighboring amino acids on ribosome only. Okay, so this is the correct one. The bring amino acid on the A side of ribosome. No, it is not possible. Translocation of the ribosome on mRNA. It is not possible. Shifting of amino acids from A side to B side. This is not also not possible. Okay, so what is the meaning of this one? We will see. Go to the next one. If adenine makes 30% of the DNA molecules, what will be the percentage of the thymine, guanine, and cytosine in it? Okay, see here, adenine, that is equal to thymine, isn't it? So here 30% means 30% must is there. 30 plus 30, how much is that? 60. Okay, remaining what is that? Guanine and cytosine. Guanine and cytosine, how much is that? 100 minus. See here, note it down, my dear students. Here they have given us a adenine, how much is that? 30%. So adenine is always equal to, okay, the adenine always equal to thymine. Okay, so thymine. So 30 means, so thymine is also how much is that? 30 is there, isn't it? Adenine is 30 given here. Adenine makes 30% of the DNA molecules, isn't it? So here I have given adenine is 30. So adenine always combined with the molecules of thymine. So it is also 30. So 30 plus 30, how much is that? 60, isn't it? 60 molecules. Okay. 60. How much percentage means 100, isn't it? 100. 100. Is that one? 100 minus 60, how much is that? Equal to 40. So this one indicate which one, guanine and cytosine, isn't it? So the answer is, what is that, Answer it. What is that? See that one? So this is the correct one. Okay. So you will get, how much is there? 40. 40 means you have to split into 2. Depends upon the, what is that? Combination. So how much is that? Guanine is how much is that? 20% plus 20. So total how much is there? Just imagine, guanine is 20 and also cytosine is 20. Here Adenine 30 is the, already know 30% means, and adenine always combined with the molecule of thymine. So it is also 30. So 30 plus 30, 60 is the percentage means 100. You minus the adenine and thymine combination, you will get 40. This is the, what is that? Combination of which one, guanine and cytosine. So 40 divided by 2 codons, so you will get it 20 plus 20. So this is the correct answer. So the answer will be 3. Okay. Are you clear, my dear? Ajay?
Yes, sir.
Okay. So any doubt in this part, my dear? Any doubt in this
No,
part?
sir. No, sir.
Okay. Go to the next one. So this is the third one. Fourth one. Already you know about, already we saw it. The match the following genes. I genes. Note it down, my dear student. This is very, very important. I is denoted as a repressor genes. Okay. Jet is the first one. So it comes under the heading is known as, it's a jet gene produce the beta galactosidase. And also A gene, trans-acetylase. And Y gene is a permeases. Okay. So you know about this point already. So the answer will be which one, fourth one. 3, 1, 4, 2. Okay. This is very, very important, my dear student. Don't forget, this question must be in 2023 and 23, 4 and 5 and like that. Without this question, there is no, what is that? NEET paper, NEET question. Okay. So last one, see that the splicing process. The splicing process mostly occurs in where? Occurs in prokaryote. Join intron together. Can produce multiple mRNAs from the same transcript. Only join exon for each gene in one way. So these are the 4 options has given here. Occurs in prokaryote? No. Join introns together? No. Can produce multiple mRNAs from the same transcript. This is the correct answer he has given in your book. Only joins exon. Here the intron, here the exon. For each gene in one way, it is not possible. The answer is which one, the third one only. So this part is completely finished, my dear students. This part is completely finished. 8th one. We'll pass on to the next one is 9th one after Wednesday. Today, what we discussed, please summarize all those things. Okay. What is by genetic code? Who discovered genetic code? George Camo. How many amino acids there? 20 type of amino acids also there. How it will be divided? So no, depends upon the what is that? Genetic code only. What are the genetic code present in it? Properties of genetic codes. These are all are indicated that triplets in nature. For example, you know about the triplets codons. Okay. And also the particular amino acids. The triplet codon is also mentioned in the particular amino acids, and only one type of amino acids. Some of the category what happened, it may be mentioned more than one amino acids. Okay. So that is the main properties. The code is nearly universal only. So universally they are following all the category, whether it may be higher animal or lower animal, whatever it may be, they are universally followed, just like ICBN. Okay. Almost all organisms in nature, forms bacteria to humans, use exactly the same genetic code only. Same genetic code only they are following. That is very, very important things, my dear. This is another one. The rare exception included some changes in the code mitochondria and also protozoans. This is the different. How many amino acids are there? There are 20 amino acids there. They are all are considered the headings 20 levo-rotatory alpha amino acids is there. Okay. Don't forget, my dear student. This is a structure of the only one amino acid. They have been given L-alanine and D-alanine. Levo-rotatory and dextro-rotatory. Okay. So levo-rotatory and dextro-rotatory. This is the structure of the amino acids. See the next slide. So you can easily identify the L-alanine, D-alanine, dextro and levo-rotatory. See the structures, the amino group. Okay. It's a left side. This side, right side. So likewise, they mentioned, okay, levo-rotatory and dextro-rotatory. So what are the types of amino acids already given? There are 2 types of amino acids that have been reported by the scientists. One is called as essential, another one is called as non-essential. Essential amino acid, it will not synthesize in your body, already know about. Non-essential amino acid will completely synthesize in your body. Okay, what are the essential amino acid means? They have given in the tabulations format. histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valines. You have to form as a mnemonics aspects. In this party, alanine, arginine, aspartic acid, asparagine, cysteine, glutamic acid, glutamine, and also glycine, proline. These are the non-essential amino acids. Okay. So both are essential for the biosynthesis of what is that? Protein molecules. Okay. And also genetic code is also how they may be formed in 64 genetic codes with the help of the checkerboard only. Already you know it's also one of the checkerboard like structures only. Okay. Suppose if it is one code is present in it, A, C, G, and U, it will not mean it not sufficient for the 20 amino acids sequences. Okay. Suppose if it is double, it is also same. It is also same. It is not sufficient because only the 16 codon will be formation will be possible. Okay. Suppose if it is 3, how much is possible? You know, triplet. Okay. How much is the triplet? So total is 64 codon is possible. Okay. This is worldwide accepted one. That is very, very important, my dear students. So that has given in the this. Okay. See that one. Okay. So this is the one. So this is the codon already. How it will be made by the checkerboard. This is the first letter, second letter. This third letter also the UCAG, UCAG. Here UCAG is you have to put in the each and one. So okay, 4 in the single column. So you have to mix with the first letter here, U, and second letter this one. Take a third letter from the here. Same things. These are all are comes to UU. See here in this UU. So you add this last letter from the last one, UCAG. In this category, you add the first letter U and second letter UC. So you add UCAG here. The same thing is also repeated again and again. Different letters. Finally, we'll get it. 64 codon is here. Okay. From this part, the starting codon is started from AUG. It is also denoted as methionine. And the stop codon in this part is that it has a specific name. Okay. That is also very, very important. Different colors also has given here. The polar, non-polar, and aromatic, and positively charged and negatively charged. Here you must note it on only one is the negatively charged amino acid present in it among the 64. Only the aspartic acid only. And another one is that aromatic amino acid. So many times they were asked the questions: phenylalanine, tyrosine, tryptophans. Okay. So these are the 3 things is very, very important. And one more is that positive charged lysine, histidine, and arginine. Okay. These are the important things. So already know all about these things also. And how it will be divided starting codon. Okay. So remaining things we will see on the next class, my dear students. Okay. Today the part 8 finished. Thank you so much for the participation. Thank you so much, my dear students. Good night. Take care. Thank you, Ajay.
Thank you, sir.
Thank you, ma'am.
Thank you, sir.
Thank you, ma'am.